All right, so essentially, guys, this is going to be the pre-lab, okay, for our microscopy lab that we're doing a week from today, okay? But of course, you guys have tomorrow and Monday off, so it's not really a week, okay? Right, the problem we're investigating here is how does a microscope allow us to see tiny specimens? All right, we talked a little bit yesterday about how Hooke's microscope worked, okay, and how it was basically just that he put a bunch of lenses in a row and it made the image bigger. Does anyone remember how that happened? What did it do to the light? It refracted it or bent it or changed the path of it as it passed through the lenses. And if you put the lenses in the right alignment, then you'll get the path of light resulting in an enlarged image. All right, and that's how microscopes work. Okay, it's essentially also how telescopes work. Okay, anything that magnifies something. All right, so we want to first off identify our manipulator, responding, control variables, and as always explain our choices. What we're doing here, guys, is that we are going to look at a few different specimens. Okay, we're going to look at a letter E that we cut out of a newspaper. Right? I know that's not terribly exciting, but the purpose of looking at the letter E is we know how a letter E is supposed to look. And if it looks different through the microscope, and I mean that is that it's not right side up, okay, maybe it's turned somehow, then that helps us to figure out exactly what's going on with the light as it goes through the lenses. Everyone follow me? Okay, if we put the letter E in, and we know when we put it in under the microscope that it's right side up, and it looks different when we look at it through the microscope, we can make some assumptions about what's going on with the light on its way to our eye. Okay, so that's the purpose of looking at the letter E. It's actually the thing that's going to let us know whether, we can solve our problem, and when we propose our hypothesis, whether it's right or not. Okay, so we're going to be looking at the letter E. We're also going to be looking at an amoeba, a paramecium, and a euglena. Okay, all three of those are protists, single-celled eukaryotic organisms. Okay, eukaryotic organisms are organisms like ourselves. Their cells have internal structures called organelles. Right, so they have a nucleus and they have mitochondria and all those kind of things. Bacteria are prokaryotic cells. They don't have those structures. They're still cells, but they don't have the internal structures. Okay, so these are eukaryotic cells. In terms of a single-celled organism, they're all quite large. Okay, and that will enable us to get some practice using the microscope and figuring out, how does it focus best and things like that. So basically just to practice with. So we're looking at those three things, and I would suggest looking at them in that order. Okay, looking at the amoeba first, the paramecium second, and the euglena last because they get smaller. Okay, the amoeba is the biggest and the most easy to find. Okay, the paramecium is also fairly large. Okay, it's relatively easy to find. The euglena is shaped a lot like the paramecium, but it's way smaller. All right, and so you have to be a little bit more proficient with the microscope in order to find that one. Okay, now we're changing what we look at. Okay, hint, hint. We're looking through the microscope. Hopefully, they what? When we look through the microscope, they'll hopefully be No, I mean, when I look through the microscope at these specimens, they will hopefully appear larger. Okay, I would say appearance might be something that I'm looking for, okay, in this experiment. Right? Controlled variables, yes, anything that remains the same. Okay, are we looking, are the microscopes pretty much all the same? Sure they are. Okay, are the magnifications pretty much the same? Sure they are. Right? Things like that. Even the technique that you use, okay, should pretty much be the same each and every time. So think about things like that. Boy, I gave you a lot there. Okay, hypothesis. So if, and then statement. Okay, the if part had best talk about that. Okay, the if part had best talk about how a microscope works. The and part, as always, describes the experiment. We're looking at some microscopic organisms. Okay, the then part should probably talk about your responding variable a little bit, and that would be, okay, the appearance. Okay, tell me what you think is going to happen to them when you look through there, especially the letter E. Okay, things like that. And is it okay if, when you write this hypothesis up, what you predict about what they look like is wrong? Is it okay? Sure it is, as long as in your conclusion you say, well, I can't accept my hypothesis because it ended up looking like this, and that means that the light did this as it went through the microscope. Okay, things like that. It's fine. Okay, I'm not going to mark your hypothesis wrong because I know ahead of time that your prediction isn't right. That's not how it works. Okay. All right. Then for the procedure, you're going to get your microscope. Okay. It says get the necessary slides, but you're only going to have one slide at your table at a time. All right. It just eliminates the possibility of sweeping one off accidentally onto the floor. Okay. If you only have one and it's in your microscope, that doesn't happen. Okay. When viewing the slides, make lab drawings of the letter E, paramecium, euglena, and amoeba. I'll show you how to do that today. Okay. Because we're actually not going to use the old-fashioned way with pen and pencil. All right. Because we're going to do this electronically. Right? We're actually going to use the cameras on your phone to take pictures through the microscope of what it is you're looking at. Okay? And that'll be a lot better than drawing, especially for someone like me who still draws people with the legs and arms coming out of the head. Okay? All right. And then each group is going to create a wet mount of the letter E, which I'll also show you how to do. Okay? And like it says here, be sure that the letter E is right side up on the slide when we're looking through the microscopes. Okay? For the analysis, obviously you're not going to be able to do that until you're done the lab. Okay, but using what you saw under the microscope, that is the size, the orientation, general characteristics, explain what you think happened to enlarge the specimen. So basically, what did the light do as it went through? Okay, describe and explain the steps you took to find and focus your paramecium. So walk me through. Okay, first I did this, then I did this, and so on. All right, and then thirdly, why do we add water to a wet mount? I'll give you a little tip on this. It has something to do with this. See, I'm not a liar. I told you that would come back. Okay, it's coming back early, right? That polar nature of water thing. Okay, and then conclusion evaluation, same as always. Okay, restate your hypothesis, explain why you accept or reject. All right. Now, what we're looking at here, okay, under the microscope, these are euglena. Okay, they're little green things. They're actually really cool in that they are mobile. Okay, they have little cilia and a flagellum. Okay, but they're photosynthetic. Okay, so they can actually produce food through photosynthesis using only light and carbon dioxide, but they can also move. Okay, so in that way they're very cool, very unique kind of organism. All right, that's kind of their general shape. Okay, the lab drawings. Okay, this is kind of the format of them, and I worked really hard to draw this plant cell, and it's still terrible. All right, but they do need to be in this format. At the top, and if you look on the sheet I gave you, turn it over, there's a proper lab diagram there on the back. Okay, format is as follows, and you do have to follow this format, okay, because when you get to university, this is the format they're going to want it in, and they're really, really anal about how you do it. If you don't do it their way, you get it wrong. Okay, and yeah, it's like as simple as I didn't, put the date on the top. All right, so you put your name up at the top, okay, and then the date right underneath it. All right, so obviously it's not 2010 anymore, but when I make a good drawing, I keep it. That's a good drawing for me. All right, then, okay, you draw your drawing, and this isn't like in art class, okay, and well, it won't matter because you're not actually going to be drawing it with pencil, okay, you're going to be taking a picture of it, but if you were to actually draw it with pencil, you don't sketch, you actually make one continuous line. But for us, we don't have to worry about the mechanics of the drawing because we're going to take a picture. Right? But what we are going to have to do with our picture is take lines out from all of the structures off to one side. All of your labels go to the same side. And the labels should all be on the end of a horizontal line. All right? What we don't want to see is a bunch of lines going all over the place, okay, kind of randomly and haphazard because it starts to look disorganized. Right? So you can see on this one, all of my lines go to the same side. Even this one, I couldn't put it over here. I couldn't go straight out from the nucleus. Okay? So I had to go down and then over. Right? That's fine as long as my label ends in a horizontal line. Sometimes things get too close together and you've got to go down and over to make room. That's okay, but they should all end in a horizontal line with the label at the end of the line. Okay? Underneath needs to be the name of what you're looking at. So oddly, the title goes at the bottom. Okay? And that's the part most people get wrong because it's counterintuitive. Okay? Title goes at the bottom. The magnification at which you viewed it. So if you looked at it on high power, that would be 400x. If you looked at it on scanning power, that would be 40x. Okay, and your estimated size. That's why we went over all that stuff about estimating the size of an object. The estimated size of the object is the last thing that goes on there. Okay, everybody with me on that one? Okay. And you guys already have that stuff, so we don't have to talk about that anymore. Okay, the wet mount of the letter E. Right? When you make a wet mount, you're going to take a blank slide. It's just a little rectangular piece of glass. Okay? And you're going to lay it on the table like so. You'll cut out your letter E, and you'll place it right here on the piece of glass. Obviously, it's not that thick, but you need to be able to see it because we're looking at it from the side. All right. Then you're going to take a little eyedropper like we used in the chem labs and drop some water on top of that. Okay. Now, when you do that, it'll probably flip the little clipping that you made over. That's fine. Just take some tweezers or your fingers or whatever and just make sure that the letter E is flipped back right side up. Move it around a little bit, okay, so that it's stuck to the slide and it's right side up. It's okay if you do that. Right? Once you've got that, so now you'll kind of have a bubble of water in your letter E. Okay, once the letter E soaks up the water, it'll sink. Okay, then you're going to take what's called a cover slip, which is a small little square piece of plastic. You do have to be careful with these. I've had people cut themselves on them. They're that thin that you can actually slice yourself pretty good if you're fooling around with the edges of them. Right? But it's a little square piece of plastic, and what you're going to do is lean it at an angle close to where your letter E and the water are, and then just let it fall. Right? So you'll let it fall and it'll come down and it'll squish the water out, okay, kind of all around the sides, and then it'll stick. Okay? The water makes it stick somehow. Now you can view that underneath the microscope. Okay? Just stick it on the stage, inside the stage clips, and start focusing. Okay? So that's how you make a wet mount. I'll review that again on Thursday of next week when we do this, but that's how you make a wet mount. All right, again, these are the specimens we're going to be looking at, okay? This is your euglena up here. Okay, this is your amoeba. And this is your paramecium. Oh, paramecium. Okay, so they all look a bit different. The amoeba, they don't always have this shape. Okay, an amoeba is, it's like a piece of jello, right? And it can move all over the place, and it has these things here called pseudopods, which are false feet. It's how it feeds. It engulfs things in its pseudopods, and then it comes inside and it eats it. All right, so yeah, it's not necessarily going to be this exact shape, but it'll be a little purple blob. All right, so you're looking for a purple, or a couple of the slides are dyed blue. They might be a blue blob. Okay, but that's essentially what they're going to look like, a blob. Okay, you're looking for a blob. Questions on those? Okay. Now, I'll show you how to do your lab drawings here with Google Docs in a minute. I'll get you guys the Chromebooks here, and then we'll get started on that. Okay, so the process for making your lab drawing, okay, is going to be something like this. You're going to take your phone, and you're going to put the camera against the eyepiece until you can see on your screen what you were looking at previously. It takes a little bit of adjusting, okay, but you'll eventually get it. You just kind of have to be patient and work it around until you see the right thing in there. Okay, then you're going to take a picture of that, and I'm just going to cheat and take a picture of what I have on the smartboard here. Okay, so I'm going to go like this and go like this. That's a great paramecium. All right, take a picture of that, and then I'm going to email it to myself, all right, from my phone, which isn't hard, right? We just go to Photos, and then we punch in, okay, who we want to mail it to. All right, so I'm just going to send it to my student account here. Okay, and if you're on the Wi-Fi while you're doing this, for some reason I'm not. Okay, you can send it to yourself big. Okay, make sure you email it to yourself with like actual size. Okay, so it's nice and big and it doesn't pixelate on you. All right, so then once I've done that, okay, I go into here, and then I should get, let's refresh here, should be there. Not yet, for some reason. Okay. So now the picture I just took, okay, has come to me, okay, and if I open that up, all right, there's my picture, and I'm just going to save that, okay, into my Google Drive here. Whoops. Oh no, where did that go? Okay. Okay, so once you've copied that, okay, just right-click on it and copy it or whatever. Okay, then insert a page break. Okay, and you can start your lab drawings here, right, and simply follow the same format. Okay, you can justify left here for the first. Whoops. Okay, so just justify to the left, and where is that? Why is that not showing up? There we go. Okay, so you can put in there, all right, here's my name, and here's the date, so it's October the 9th, 2014. All right, and then I'm just going to paste in there my picture. Yeah. Okay, so now my picture is in there, and for some reason it's rotated, but okay, you can play around with the formatting and all that kind of stuff. Then below that, you'll put in your title and all that kind of fun stuff. Oh, come on, stop doing that. Okay, so we'll go back and now we'll center it because that's what we want now for the title and stuff. All right, so we'll say paramecium, okay, 400x, okay, and it'll be, I don't know, 200 micrometers or something like that. Okay, and you can use or you can insert the symbol for it. I think they have it in here as well. But we'll do that later. Now for the labels, okay, you'll have to draw a little bit. Okay, so insert some lines. And man, I'm just having a total blank here right now. Okay, so you insert horizontal line. Okay, go down to horizontal line, and then you can draw. Okay, or actually it puts it up there for some reason. I don't know why. It's not what we want. Okay, so to do this lab drawing, you actually don't insert the image into your document directly. You insert it into drawing. So you go. I'm just going to save and close that. It's going to be ugly here, but oh yeah. Now of course the smartboard won't respond to that. Okay. Oh no! Don't do that to me. Oh, sure. Thanks a lot. There we go. Okay, so go to, we're just going to get rid of this completely here. All right, and we'll try this all again. The right way. Okay, so go to, once you're into here, okay, you go into Insert Drawing. All right, and when you're in Drawing, okay, you get this kind of canvas or whatever they call it. All right, just click Paste. Yeah, okay, don't click Paste. Click Control-V because it likes that better. Okay, it'll paste your image in there, and then you can start putting lines on that, okay, that come out to where you're going to do your labels. So basically just draw them out to the sides, shrink your drawing when you insert it back in, and then you can draw the labels down the side of the Google Doc. Okay, you can actually write the labels in at the side. So in the end, it'll look something like, okay, so I'm just going to label this thing here. It's going to come out to the side. I'll label that thing there. And it'll come out to the side. Alright, something like that. And I'll go save and close. Okay, it'll insert it like so. And then over here, I will resize this just slightly. I'll wrap my text around it. Okay, that way I can write on both sides. No, I can't. Over there. Come on already. Oh, it's got to be that kind of day. just don't want to cooperate today. Sorry, I made it too big. For whatever reason, it wouldn't let me write beside it. Okay, but now you can write beside it, and you can actually take the margins off as well here. No, it doesn't really matter. Just leave it the way it is. And you can write in the labels beside. Okay, everyone with me there?